A simple paper cut. A tickle in the back of your throat that you just know is going to ruin your week. That sudden, inexplicable wave of itchiness after trying a new restaurant. In these tiny, everyday moments, a decision of monumental importance is being made inside you. A decision that could lead to a swift resolution, a week of misery, or even a full-blown civil war. Who gives the order? Who is the ultimate authority that decides whether your body should unleash overwhelming force, meticulously repair the damage, or wisely, do nothing at all? You might picture a single, 5-star general sitting in a biological command center, looking at a map and decisively pushing a red button. The reality is infinitely more fascinating, more complex, and frankly, more chaotic. Your body doesn't take orders from a dictator. It doesn't have a king. Your body holds a debate. Your immune system is a sprawling, brilliant, and sometimes flawed democracy. It is a parliament of trillions, a vast society of cells where microscopic agents cast biochemical votes, present compelling evidence, lobby for their own interests, call for backup, and together, through a complex and beautiful consensus, they choose a path. Kill, heal, or stand down. Welcome, citizens, to the microscopic nation that you inhabit. To truly understand its politics, you first need to get acquainted with its two main houses of parliament. Think of them like the House of Representatives and the Senate. Each has its own culture, its own speed, and its own unique powers. First, there's the House of Innate Immunity. These are your first responders, your frontline infantry. They are breathtakingly fast, brutally blunt, and they have absolutely no time for nuance. They are the patrol cops on the beat, the bouncers at the club door. They aren't looking for a specific criminal's face. They are trained to recognize the general signs of trouble itself. Scientists call this pattern recognition, and it is one of the oldest, most evolutionarily conserved parts of life on Earth. What are they looking for? Two telltale signatures of chaos, PAMPs and DAMPs. PAMP stands for pathogen-associated molecular patterns. Forget the complicated name. Just think of these as the universal, unchanging signatures of invaders. It's the chemical equivalent of a skeleton key, a ski mask, or a crowbar. It's the unique texture of a bacterial cell wall, the strange double-stranded RNA that only viruses produce. Your innate cells have receptors, like molecular motion detectors called toll-like receptors, that are perfectly designed to spot these patterns. When a PMP is detected, the cell doesn't need to know if it's Staphylococcus or Streptococcus. It just knows it's a bacterium, and that's enough to sound the alarm. Then you have DAMPs, damage-associated molecular patterns. This is the alarm signal of internal crisis. It's the smell of smoke when your own house is on fire. When your own cells burst open from injury, stress, or lack of oxygen, they release molecules that should always be inside. Things like ADP, the energy currency of the cell, are suddenly floating around outside. To an immune cell, this is the molecular sound of shattering glass. It's an unambiguous signal that something has gone terribly wrong. When the innate system's detectors, these toll-like receptors, light up with either PAMPs or DAMPs, it doesn't form a committee. It doesn't wait for permission. It acts immediately. The first on the scene are often the neutrophils. Think of them as the cellular SWAT team. They are short-lived, fanatical killers that swarm an area, release powerful chemical weapons, and die in the process. Then come the macrophages, the heavy-duty cleanup crew and interrogators. They engulf pathogens, eat dead cells, and start releasing their own signals. A general state of emergency is declared, inflammation. That redness, that swelling, that heat, that is the sign of your innate democracy rushing resources to a crisis zone. But this system, for all its speed, has a weakness. It's generic. What happens when the threat is a sophisticated virus that hides inside your own cells? Or a novel pathogen the body has never seen before? For that, you need the second, more deliberative chamber, the Senate of Adaptive Immunity. This house is slower. Much slower. Days, even weeks, slower. But it is astonishingly powerful and surgically precise. It doesn't just recognize trouble. It learns the enemy's name, face, date of birth, and shoe size. Then, it designs a custom weapon to neutralize that one single threat and remembers it for the rest of your life. This is the domain of your T cells and B cells, the elite detectives, the criminal profilers, the special forces snipers of your body. They are generated through an incredible process of genetic shuffling called V-D-J recombination, which creates a standing army with millions of different receptors, ensuring that no matter what new shape an enemy takes, someone in the army will be able to recognize it. Now, these two chambers can't work in isolation. A functioning democracy requires communication. The frontline cops need to be able to talk to the elite detectives. And their primary meeting halls, their bustling intelligence hubs, are your lymph nodes. You've felt them, swollen and tender in your neck or under your arm when you're sick. That isn't a sign that the sickness is winning, it's the sign that your government is in a critical, emergency legislative session. The lights are on all night, and the debates are raging. Let's follow one piece of intelligence through the system. Picture this: out on the frontier of your skin, a dendritic cell is quietly surveying its territory. Think of it as the ultimate intelligence agent, a sentinel. A splinter introduces a new, unknown bacteria. The dendritic cell's PMP receptors go wild. It becomes activated. It engulfs the invader, drags it inside, and brutally dismantles it for parts. It then takes a small, unique piece of that bacteria, a protein fragment called an antigen, and displays it on its surface using a special molecule called MHC. The dendritic cell has just created a wanted poster. Its mission now changes completely. It stops being a sentinel and becomes a messenger, the Paul Revere of the immune system. It pulls up stakes and begins a journey, migrating through your lymphatic vessels until it reaches the nearest lymph node. It arrives at this bustling convention center, filled with millions of T cells, and it bellows, "I have urgent intelligence from the front lines." This process, antigen presentation, is the sacred moment where the innate and adaptive worlds connect. It is the beginning of the real, high-stakes debate. Inside that lymph node are hundreds of millions of T cells, each one a unique delegate. Each T cell has a receptor on its surface, a unique lock, that was randomly generated. Each lock is designed to fit only one specific key, one specific antigen. So, as the dendritic cell pushes its way through the crowd, holding up its wanted poster, it bumps into T cell after T cell. Nothing. 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 Then, out of a million cells, one T cell's receptor fits the antigen perfectly. The key slides into the lock. Click. A connection is made. A shiver of recognition runs through the cell. But stop right there. A single click is not enough to launch a war. And thank goodness for that. If it were, your body would be in a constant state of civil war, attacking harmless food proteins, friendly gut bacteria, and even your own cells. To prevent this catastrophic chaos, your immune system has evolved an elegant constitutional safeguard. It is an absolute, non-negotiable law, the 3-signal rule. Signal 1 is the match we just described, the T cell receptor, the lock, clicking with the antigen, the key. This tells the T cell what the potential enemy looks like. It establishes identity. The T cell now has a target. But it still has no context. Is this target a real threat, or just a piece of harmless debris? To answer that, it needs signal 2, co-stimulation. This is the crucial confirmation, the two-factor authentication of immunity. Remember how our dendritic cell was first activated by PAMPs from the bacteria? That initial danger signal caused it to put on a second molecule, a protein called B7. The T cell, in order to become fully activated, must use one of its own receptors, called CD28, to shake hands with the B7 protein on the dendritic cell. This handshake is a security check. It confirms that the antigen being presented isn't just some random molecule, but that it was collected from a genuine crime scene, a place with signs of damage or invasion. Without this second handshake, the first signal is dismissed as a probable false alarm. The T cell enters a state called energy. It is alive, but rendered politically silent, unable to vote or participate. This is how you avoid attacking the piece of steak you just ate. The protein antigens are presented, but without the DAMPs or PAMPs, there's no B7, no handshake, no activation. Okay, so now we have 2 signals. The T cell knows what to attack and has confirmed that it is genuinely dangerous. The vote to act is gaining momentum. But there's one final, crucial question to answer. How should we act? What kind of war do we need to wage? A bombing campaign? A ground invasion? Or a diplomatic mission? That brings us to the all-important signal 3, the cytokine milieu. Cytokines are the ballots of the immune system. They are tiny signaling proteins that cells release to communicate with each other. The specific mix of cytokines in the lymph node at that moment creates the political atmosphere in the chamber. It's the weather report that tells the T cell what kind of battle is brewing. Is the room flooded with interferon gamma and IL-12? This is a hawkish, aggressive environment that screams, the enemy is a virus or an intracellular bacteria, hiding inside our own cells. We need to activate the assassins. We need drill sergeants. A T cell activated in this environment will differentiate into a T helper 1 cell, or Th1, a commander specializing in cell-mediated immunity. Or maybe the air is thick with IL-4 and IL-5. This is a completely different political climate. It whispers, the threat is a parasite, a worm, or a major allergen. It's too big to eat. We need to expel it. Mobilize the exterminators and bomb disposal squads. The T cell now becomes a T helper 2 cell, or Th2, an expert in orchestrating antibody responses that tag and neutralize large extracellular threats. What if the signal is IL-17? That's the call of the wall guards. It signals a fungal or bacterial invasion right at the barriers, the skin, the gut. The T cell becomes a Th17 cell, dedicated to recruiting those SWAT teams, the neutrophils, to reinforce the barricades. And finally, what if the dominant message is one of peace? A cytokine cocktail rich in TGF-beta and IL-10. This is the call for de-escalation. It says this situation is delicate. An overreaction would be disastrous. We need restraint. We need diplomats. The T cell now becomes a regulatory T cell, or TREG. Its entire job is not to fight, but to keep the peace, to travel the body and tell other, more aggressive T cells to stand down. The 3 signals have been received. The vote is in. The T cell has been given its mandate. Now, the consequences of that vote unfold. If the collective decision was kill, the system mobilizes with a quiet, chilling ferocity. The newly minted Th1 cells begin cloning themselves, creating an army of commanders that spread throughout the body. They find macrophages and supercharge them. But the real stars are the cytotoxic T lymphocytes, or CTLs, the true assassins of the adaptive system. They hunt down any of your own body cells that have been infected, recognizing them because the infected cell displays the telltale viral antigen on its surface. The CTL latches on and delivers a kiss of death. It uses a protein called perforin to punch a hole in the target cell's membrane, and then it slips in a package of toxic enzymes called granzymes. This is not a bomb, it's a coded self-destruct note. The infected cell receives the note and dutifully initiates its own controlled demolition, a process called apoptosis. It's a clean, precise, cellular drone strike that eliminates the enemy's factory without causing excessive collateral damage. If the vote was heal, the entire tone of the battlefield shifts. The war is over. Reconstruction must begin. Th2 cells and other signals encourage the macrophages to switch from their aggressive M1 demolition crew persona to their peaceful M2 gardener and construction foreman mode. They stop spewing chemicals and start releasing powerful growth factors. They call in fibroblasts to lay down a new collagen scaffold. They meticulously manage the cleanup of cellular debris. The inflammation recedes. It's a beautifully orchestrated transition from total war to total reconstruction. And what if the vote was stand down? This is not a passive process. It is just as active and critical as the others. Peace must be enforced. The system has multiple ways to do this. T cells that have completed their mission, or which are perhaps too aggressive, are instructed to retire via that same apoptosis process, triggered by a specific handshake known as phosphosel. Other cells are pushed into that silent state of energy. And most importantly, those diplomatic TREG cells circulate as a permanent peacekeeping force, releasing their calming cytokines, IL-10 and TGF-beta, reminding everyone that tolerance is an active policy, not a sign of weakness. While all of this is happening with the T cells, a different, even more dramatic election is taking place in another part of the lymph node. It's the B cell election, and the prize is the creation of the perfect weapon. B cells are your body's antibody factories. But you don't want a generic, one-size-fits-all antibody. You want a bespoke, high-precision guided missile. So, in specialized structures within the lymph node called germinal centers, think of them as a brutal, hyper-competitive military boot camp. The B cells that have recognized the antigen begin a frantic process. They activate a special enzyme that intentionally introduces typos, or mutations, into the genes that code for their antibodies. This is called somatic hypermutation. It's an incredible evolutionary gamble. They are creating thousands of slightly different antibody designs, hoping that one of these random changes will make the antibody bind to the enemy even more tightly. Then, the competition begins. The B cells present their new and improved antibody designs to a special kind of T cell, the T follicular helper cell, which acts as a ruthless quality control inspector. Only the B cells whose antibodies bind with the absolute highest affinity get a survival signal. The ones that perform poorly, or whose antibodies might accidentally recognize a self-antigen, are commanded to self-destruct. It's a brutal process. The handful of winners from that round then go back, mutate their genes again, and compete in a new, even more difficult round. Iteration after iteration, a champion is selected, a B cell that produces an antibody of breathtaking affinity and specificity. Some of these winners will become plasma cells, which are living antibody factories, churning out millions of these perfect weapons into your bloodstream. Others will become memory B cells, the quiet archivists who store the winning blueprint for decades, ensuring that if this enemy ever returns, the response will be immediate and overwhelming. So you can see, this entire system is far more sophisticated than the old, simplistic idea of self versus non-self. That model is outdated. The immune system is much smarter than that. It predominantly follows what immunologist Polly Matzinger famously proposed as the danger model. It doesn't just ask, "Are you one of us?" It asks, "Are you causing trouble?" The trillions of bacteria living peacefully in your gut are entirely foreign, but your immune system actively tolerates them because they arrive without danger signals. They don't break any windows. They are good citizens. But a sterile injury, like a burn, contains no foreign invaders, yet it triggers a powerful immune response. Why? Because your own dying cells release those DAMPs, those danger signals. The system is responding to the context of danger, not the mere presence of foreignness. It's like a sophisticated car alarm. It doesn't go off because a person with a foreign accent walks by, non-self. It goes off because someone smashes the window, danger. But as we all know, even the most well-designed democracies can be messy. Governance can fail. Procedures can be hijacked. And when the immune democracy fails, the results are some of our most challenging diseases. Autoimmunity is what happens when the votes are catastrophically miscounted. The checks and balances break down. A T cell that recognizes a self-antigen somehow gets both signal 1 and a mistaken signal 2. The parliament declares war on its own loyal citizens. In type 1 diabetes, the T cells mount a deadly attack on the insulin-producing beta cells in the pancreas. In rheumatoid arthritis, the immune system targets the lining of the joints. It is a tragic case of friendly fire. An allergy is a wild, hysterical overreaction to a harmless piece of legislation. A pollen grain, a particle of dust, a protein from a peanut, these are presented to the parliament. Instead of being correctly identified as harmless and ignored, they trigger a full-blown Th2 emergency response. Your B cells are told to produce a specific class of antibody called IgA, which attaches to mast cells. The next time you encounter that pollen, it cross-links the IgA on your mast cells, causing them to degranulate, to release a flood of histamine. That is the runny nose, the itchy eyes, the constricted airways. It's the fire department flooding the entire building because someone lit a birthday candle. Cancer is perhaps the most insidious failure of governance, a story of treason and exploitation. Cancer cells are your own cells, but they have gone rogue. And they use their insider knowledge to exploit the rules of the chamber. Many tumor cells learn to cover their surface with a very specific badge, a protein called PD-L1. Your T cells have a receptor called PD-1, which acts as a natural off switch, a brake to prevent excessive immune responses. When a T cell arrives to investigate the tumor, the cancer cell shows its PD-L1 badge. The two proteins bind, and it sends a powerful stand-down signal directly into the T cell. It is a procedural veto. The T cell, even though it may recognize the cancer as dangerous, becomes exhausted and walks away. The traitor is hiding in plain sight, wrapped in a stolen flag of tolerance. This discovery, by the way, led to a Nobel Prize and the creation of checkpoint inhibitor drugs, which work by blocking this treacherous handshake. And then there is the cytokine storm. This is what can make infections like severe influenza or COVID-19 so deadly. It is the immune system's equivalent of emergency powers without any checks and balances. A positive feedback loop ignites. A few activated cells release alarm cytokines like TNF-alpha and IL-6. These cytokines then trigger more cells to activate and release even more of the same alarm cytokines. The debate chamber descends into a screaming, panicked mob. The resulting systemic inflammation causes leaky blood vessels, plummeting blood pressure, and organ failure. It is a terrifying example of a government whose response becomes far more dangerous than the crisis it was trying to solve. But here's the most hopeful part. This system is not static. It can learn, it can adapt, it can be taught. We've discovered that even the innate first responders have a form of memory, a concept called trained immunity. After an infection, certain innate cells like macrophages can be left with epigenetic bookmarks, like chemical sticky notes left on their DNA, that change how they respond in the future. It allows them to react faster and stronger the next time, even to an entirely different threat. This helps explain why some vaccines, like the BCG vaccine for tuberculosis, seem to offer broader protection than just against the single disease they target. A vaccine is not just a wanted poster, it is a profound civics lesson that can actually rewrite parts of your immune constitution. And we are not alone in this. Your microbiome, that bustling, trillion-member inner city of microbes in your gut, is constantly, actively lobbying your government. When you feed them well with fiber, they produce metabolites like butyrate. This butyrate is like a massive campaign donation to the Party of Peace and Tolerance. It directly sponsors the creation of those diplomatic Treg cells. Your diet is, in a very real sense, a foreign policy decision that shapes the political landscape of your gut, which in turn profoundly influences the temperament of your entire immune democracy. So, what does this all mean for us, the person who is the living, breathing nation-state in which this all takes place? It means we have more influence than we might think. We are not helpless observers, we are the president. Our executive decisions shape the political climate. Chronic stress is like a relentless, 24/7 cable news channel screaming about crisis, biasing every single vote towards panic and inflammation. The stress hormone cortisol can suppress your thoughtful adaptive Senate while putting your reactive innate house on a permanent hair trigger. Sleep is the parliamentary schedule. It is the sacred, off-the-record time when the day's intelligence is processed, memories are consolidated, and waste products are cleared out. Deep sleep is absolutely essential for forming robust, long-term T-cell memories after an infection or a vaccination. To rob yourself of sleep is to ask your government to make life-or-death decisions while it is sleep-deprived, disorganized, and drowning in its own metabolic garbage. Exercise is infrastructure and logistics. It ensures the roads are clear, flushing lymphatic fluid and moving immune cells efficiently through their circuits. It keeps surveillance high and response times low. And diet, as we've seen, is how you fund your lobbyists. A diet rich in diverse plant fibers is a stimulus package for your microbial allies, who in turn lobby for peace, order, and tolerance. A diet high in processed foods can fund the microbial equivalent of gangsters and rebels, who lobby for chaos and inflammation. We started this journey by asking a simple question: who decides? And the answer we found is as complex and beautiful as life itself. Everyone decides. The decision is a consensus, a wisdom of the crowd. It emerges from the chatter of trillions. From the innate chamber's primal alarm bells and the adaptive chamber's precise, custom legislation. It is a government that abides by a three-signal constitution that is swayed by the shifting winds of cytokine ballots, and that elevates the best ideas, the most effective antibodies, through the crucible of a fierce election. When this governance fails, it leads to disease. But when it works, it is the definition of health. It is precise violence, followed by careful healing, followed by graceful, enduring peace. It is the process of life, moment by moment. So, the next time you feel your body fighting something off, that fever, those aches, that fatigue, take a moment to remember the parliament within you. It's not just a mindless battle, it's a vibrant, complex, and deeply intelligent debate. And the choices you make every single day help to set the agenda and influence the outcome. To see how your body's incredible government deals with a completely different kind of threat, a threat from within, check out the video on your end screen now. And I have a genuine question for you, learning all of this, seeing your body as a democracy you preside over, what is one small thing you might do differently today to support your own inner government? Let me know in the comments below. Reading your thoughts is one of the best parts of making these videos. And if you want to continue building a deep, foundational understanding of the science that makes you you, please subscribe to the Science Audit. We're committed to creating a library of clear, thoughtful, and in-depth knowledge. Thank you for thinking with us.